Okay, so the next step is to make the sequencing library, and what we're using here is the field sequencing kit. And this is a lyophilized version, so these have been freeze-dried, and it comes with 3 tubes. There they are, tubes 1, 2, and 3. And so we're just going to put our DNA, open this up, this is what it looks like. Look at that. A little silica gel we can get rid of. It's in the same order, you can see here, small to kind of medium to large. And we're going to put 10 microliters of our DNA into the first tube, tube number 1. I'll slide this back under here. What you have to do is break that off of there like that. Tubes 2 and 3 I'm going to leave in the tray here for later. I like to go ahead and make a hole here so that I'm not jamming my tip through with the DNA into this little solution here. Okay, so 10 microliters of DNA from my aphid. There we go. You come see it mix a little closer, and I'm mixing probably gently, looks like about 5 times up and down. This is supposed to go room temp. It's not really room temp out here. It's overcast and starting to get a little cold. Field sequencing kit, you can do it room temp, or you can put it at 30 degrees Celsius, which is actually a little above room temperature. Room temperature would be 23 Celsius. I have right down here a PCR machine with a block heater that you can see from the side. I'm going to go ahead and put it inside of this block, and I've got a program cycled on here, number 400. And that number 400, what it does is it's going to heat up to 30 degrees Celsius, and it's going to run for 1 minute once it gets to the target lid temp, which is 109. So I probably put it in 15 seconds too early, but that's okay. That's how it goes. So we're going to let that incubate here for 1 minute. And then what's going to happen after 1 minute at 30 degrees Celsius, it's going to ramp down to, or up to 80 degrees Celsius. And it's going to hold at 80 degrees Celsius for 1 minute. After it goes to 80, then I have it set after that 1 minute to cool down. And I'm going to take that 10 microliters that cools down onto the next step. I'm going to take that 10 microliters and put it in tube 2, which is here, and I'm going to mix it there. So we'll stop here for a second until we get to that step. Okay, this has cooled down. It's gone through the 30 degrees and 80 degrees. We still have our 10 microliters in there. I'm going to now take that out and pipette it into tube number 2, which I've already made an opening for. This is 10 microliters. And just like before, I'm going to pipette up and down gently. The first step, what it does is that's where the DNA is cut and an adapter is attached. Now what's happening during this step is there's a sequencing adapter attached, going to be attached to that. So that's what we just pipetted into, and that's going to take 5 minutes for that to happen. Let me get my other tubes out here. What I want to do next here, and go ahead and set up the other tubes, tube 3, is I've got this set to 65 microliters, and I'm going to put this buffer here, so it's called the resuspension buffer. I'm going to put 65 microliters of the resuspension buffer into tube number 3. So that's resuspension buffer there. And I didn't make my hole for tube 3, so I'm going to do that on the fly here. There we go. I'll go ahead and put that in there, and I'm going to mix that up. This one I don't have to worry about being gentle. It's got some loading beads and sequencing buffer inside of it. And there we go. So that's mixed up. So that's going to wait, the one on the tube number 2 has to wait 5 minutes to get that sequencing adapter on there. The other thing I want to do is put these two into this tube. This is the flush buffer and the flush tether. So I'm going to put 117 microliters of flush buffer and 3 microliters of flush tether into that tube, because that's going to go into the flow cell. So I'm going to go ahead and do that right now. 117 of flush buffer. Always look at your samples before you put them in. Make sure you're getting the right volumes. Put that down there, maybe we'll put it up here. And I need, turn that off. I need 3 microliters of the flush tether for this field sequencing kit. So it looks like that. Small bit. Turn off to the side. These are supposed to be maintained on ice. So I have over here on your right, I have a ice box where I can put these things back into. So that's ready, the flush buffer, and that's got to go for at least 5 minutes to get ready there. I'm going to put these back onto ice so I can use them with another one of these. They don't go to waste. And so at this point, what I need to do next now is get the genome sequencer ready. I need to set up the flow cell and give you instructions for that, but I need to get the genome analyzer ready for that sample right there so that I can run it.